Hey, Bridget.
Hi, Melody. How's it going?
Good. How are you?
Okay, thank you.
Today was less
Today was
stressful.
good. Everything got off okay. It looks like it's running great.
Yeah, but the funny thing is, I don't know why the link I have in my calendar is still the one that you need to register.
Yes, I have the same issue. I didn't understand that either.
It's strange, right? Because I'm
Yeah,
pretty sure the link that Leah sent us is the host link. It's very
yeah,
strange.
it is very strange, but I think we're gonna have to do this each time. But it doesn't matter as long as it works.
Oh my God. Maybe what I should do is I should just like change that link or something. Maybe I'll update the link in my calendar. Maybe that would help,
Oh, that's a good idea. I can do the same
right?
thing. See if that helps.
Let's see. Post soon. Yeah, because the link that you got is that like that's the one from Leah, right?
Yes, this is the one from Leah.
Yeah, invites you to join a Zoom meetings as alternative host.
Um,
Oh,
it still says registration required. I wonder
that is
why that
so
is.
funny. So if I copy the link from there, I wonder if that will help. Um, yeah, maybe I just need to set up my own. Let's see. It's very weird. Maybe that's just what. Huh. Somehow that is simply. Maybe that's what everybody gets instead.
Yeah, I think so.
Yeah, it's weird. So really the correct link is the start meeting link for us.
Right. I think we can ask Sean. And maybe the best thing to do is to ask, I can email Sean quickly and ask him what we should do. Let me do that.
No future events, and if I just do this one, repeat week. I hope the speakers have the right link.
Has Leonid?
No, I hope they
No.
have the right link.
Oh, okay. I can send them the link quickly as well. Should I just email everybody quickly? Maybe I can
Sure.
do that.
Oh my God.
I didn't realize that Leonid wasn't on yet. Okay.
Leonid and Yi Ying.
Okay, Yi is here. I can see.
Oh, Yi is here. Oh yeah,
Okay. Hi,
great.
Yi. That's great. Do you know if Leonid has the correct link?
Hi, Yi, nice meeting you. I was starting to worry that because I want to help you guys set up to make sure the talk goes smoothly.
Yeah, I'm making sure that Leonid has the right link.
Oh,
I'm his
hi.
assistant.
Thank you so much.
Great. Yeah,
Thank you, Kelly.
Okay,
Thank
I'll
you.
unmute right now.
Okay.
Great, thank
Yi,
you.
did you want to see if you could get slides up? Do you need to practice anything?
Yeah, I can give it a try.
Okay, great.
You can try to share screen and then try to advance your slides just to make sure everything looks good.
Uh-huh. Can you see the slides now?
Yes, we can see that perfectly.
Yeah, and then you can try to do that. There you go. And
That
see.
looks perfect. Oh, that looks great.
Okay, looks great.
I think you're all set then. That's awesome.
Yeah.
Okay.
And also quick question. So Yi, you're going first, right?
I think
Or
that's what Leonid said, but I don't mind either way.
Okay, all right. Somehow you were first in the announcement, so I wasn't sure if it's intentional or.
Yeah, I think Leonid said like because I'm introducing the technology, so yeah, so he thought it makes more sense for me.
Okay, perfect.
I will also introduce you two in that order. That's why I was.
Okay, yeah, great.
Great, thank you.
So so it's like 20 minutes for the talk. Is that okay?
Yeah, that's perfect. Because in that way, you know, usually the audience is very enthusiastic and interested, so there'll probably be a lot of questions.
Yeah, you should get a lot of questions.
Oh, okay,
And you
I hope
guys
not.
can handle the questions together. You don't have to do it on your own.
Yeah, that's true. You guys can even handle
together.
the questions together. That's a good point.
Yeah,
So
you can You could probably take questions at the very end after Leonid as well if you want to. Would you prefer to do that?
Either way, yeah.
Okay, and sometimes people will interrupt you and ask questions in the middle, so just to be prepared. It's a little weird on Zoom, but we'll try
Okay.
and help with that.
Okay, yeah, thank you. Yeah, Leonid is here.
Okay,
Oh,
great.
great.
Oh, fantastic. Okay, great. We're all good then. Leonid, did you want to practice your slides? Do you want to make sure you can share?
If you want to share, and then try to advance your slides to make sure it goes smoothly.
All right. I muted myself, right?
Okay, good.
I've managed to avoid giving a Zoom presentation so far, so this is going to be
That's
interesting.
okay.
But all right, let's see. I did play with it yesterday, so hopefully. So does that work?
Yes, we
Yeah,
can see
that
that.
looks good. And
Yeah, just
maybe
so you can
try
see that.
try to do the presenter view and try to advance your slides. Sometimes that's an issue on Zoom.
Good.
Great. Looks good.
Yeah, perfect. Okay.
Looks like everything is mostly on the screen and stuff.
Yes, it looks
Yeah.
absolutely perfect.
Okay. All right.
Okay.
I'll stop sharing. I can even do
Awesome.
that. Hey.
Great. And then we usually give people a few minutes so they trickle in.
Except now,
And
now I can like see my slides, but they're still in full screen view.
yeah, so we can't see them right now.
Oh, there
so
we
we're
go.
not
Okay.
sharing, so you're fine.
Yeah. Once you
And,
start sharing, yeah, we won't see them.
And we'll have.
All right,
is
cool.
You're going to go first? Is that what was the decision? Was for you to go first?
Yeah, I think so, because she's talking about the technology, and then I talk about what we did with it. So I thought that made the most sense.
Perfect.
Great.
And I was just saying, probably most of the questions will come at the end, but there might be a few questions in between, and Melody and I as well can try and moderate and help with that.
Yeah, right.
Hopefully it'll go.
Perfect.
Yeah.
I don't think I can like talk and watch the chat at the same
No,
time.
no.
Oh yeah, no. So
sometimes what people do is they turn their camera on, they start asking a question in the middle. So you can also say, please can you hold your question to the end if it's bothering you and you don't want to answer it at that point, that's totally fine. Or you can answer it in the middle, that's fine.
Okay, yeah,
But
I mean, I'm usually totally fine
the
doing that, and like,
held till the very end, so don't worry about that.
yeah, I mean, in like real life talks, I'm totally comfortable fielding questions in the middle and all of that stuff. I'm just not sure what it's all gonna look like over Zoom.
yeah,
I'm sure
it
it'll be
can
great.
feel a little awkward, like you're talking to yourself.
I know, to a screen, right? It's like great.
But I think it'll go really well.
Maybe I'll enlist Hillary to sit across from me, so I'm like talking to an audience.
Yeah. So usually me and Bridget, if we see any questions in the chat room, we will find a good stopping point and read out those.
Yeah.
So don't worry
Okay.
about that. But if you guys prefer, we can make an announcement at the beginning that we can wait for questions at the end of both of your talks, so you guys can tackle the questions together if you like that.
Yeah, and I mean, I'll have James, who actually did all the analysis, and maybe others from the group will probably be on the call, so they can jump in. They can jump in too. Yeah, you guys decide. I mean, if somebody has something that seems, I mean, you guys know who is who and stuff, and how likely what the prior is on the informativeness of their question, right?
We're also learning. I think we're getting a hang of it over time.
We all are. All right. I'm going to take a 30-second break and get myself some water, and then I'll be...
Perfect.
Oh, and Bridget, I was wondering if you can help me during the seminar. I think last week I tried to turn off everybody's videos and sound if they forget to turn it off themselves.
Okay, sounds good. We can remind people too to turn off their videos and
now we can take charge. Oh, somehow you're not the co-host. Let me make you the co-host.
Okay.
So then you can also do that.
Okay, great. Thank you.
So Yi, when I was looking at both yours and Leonard's CV, I thought it was very exciting that all three of us came from, or we've done some of our training at UW.
Oh, I see. Yeah.
Yeah.
Yeah. I just moved in March.
Yeah, no, I saw that you just started in 2020. That's exciting.
Yeah. Yeah.
Yeah. So you did your postdoc at UW, right?
Uh-huh. Yeah.
Engineering sciences. I did my PhD in microbiology at UW.
Oh, nice.
Yeah, but I was actually at Fred Hutch. So I saw
Yeah.
Leonard actually used
Yeah,
to.
Leonid was in at
Yeah.
the Hutch. Yeah.
Yeah, small world.
Yeah.
Yeah.
Little audacious, but that's what I'm thinking. Anyways, okay, I got Mr. Now, the UPS is almost there.
Just a reminder, if everyone can turn off their sound and their video, that'll be great. Okay, and then I think I'll start the introduction maybe in a minute.
And what we will do is we will turn off people's sound and video if they don't do it when we start the talks, just as a warning.
I apologized last time.
Yeah. Yes, apologies ahead of time.
Yeah, I apologize in the chat room. Okay, great. Okay, I think I should start. Okay, great. Welcome everyone to another COVID seminar. Today we're very excited to have two great speakers, Dr. Yi Ying from the Department of Human Genetics and Dr. Leonid Kruglyak. I'm sorry, I totally butchered your name, Leonard. Also from Department of Human Genetics. So Yi, she got her PhD in genetics and genomics from Duke University, and then she was the Damon Runyon postdoctoral fellow at Harvard University, followed by another postdoc at University of Washington, Seattle. And she actually just came to UCLA, was recruited to human genetics, and just started her lab March, right, you said, this year, as an assistant professor. And when she was a postdoc in Jay Shandur lab at UW, she developed this very innovative single-cell seq method called Sci-L3 that combines combinatorial indexing and linear amplification that improves on conventional methods. And really, in terms of uniformity and through high throughput, really had great advantages over the traditional approaches. And I believe that she will also talk to you about some of the sequencing she's done on COVID today. Our second speaker, Leonard, it's very intimidating to introduce Leonard because his CV goes on and on. I will try to be succinct, but Leonard is a very well-rounded and accomplished scientist with training in multiple fields. He got his PhD in physics from Berkeley. Then he did training in natural science at Princeton. He was the NSF NATO postdoc fellow in theoretical physics at Oxford University. Then he went to do a postdoc with Eric Lander at Whitehead Institute. And there he developed this revolutionary new algorithms for linkage analysis and incorporated in a widely used computer program called Gene Hunter, which I thought it was a really cool name. And obviously, along with many other accomplishments, he was then a professor at Fred Hutch and University of Washington, Seattle. Then he went to Princeton, and UCLA was fortunate to recruit him here in 2013. And he's been the chair of human genetics since 2016. It's very impressive that he's been the Howard Hughes investigator since 2008. And he's also the founding advisory board member for bioRxiv. His lab studies the genetic basis of phenotypic variation, and over the past 25 years, he's made so many fundamental contributions that really revolutionized our thinking on linkage analysis, SNPs, and genetics of global gene expression, also called EQTL analysis. And his papers have been cited over 50,000 times to date, which is really, really impressive. And he's also a great mentor and trained many successful students and postdocs with careers in academia. So with that introduction, please take it away.
Okay, I'll start sharing. Thanks so much for the nice introduction. Okay, yeah, thanks, Melody, for the nice introduction. First, I'd like to thank the opportunity to talk. Today, I'll talk about a new high throughput and cost-effective viral genome sequencing method we've developed called V-seq for whole genome sequencing of SARS-CoV-2. So SARS-CoV-2 is a single-stranded RNA virus. The genome is about 30 kb. RNA virus sequencing can be performed by metagenomics approach, which involves constructing bulk RNA-seq library, depleting abundant human ribosomal RNA, and sequencing the rest of the RNA molecules. These include SARS-CoV-2, the virus of interest, other respiratory RNA pathogens, and host transcriptome. Alternatively, we can enrich the viral genome of interest, which is more cost-effective in terms of reducing the amount of sequencing required to capture strain-specific viral reads. Basically, you go through these steps to get all the RNA molecules first, and then add a few extra steps to enrich for the viral reads. V-seq, the method we developed here, has a much shorter workflow, such that we use targeted reverse transcription to enrich for viral reads at the very first step. The new methodology makes it both cheap and fast. I'll walk you through the workflow and how it compares with commercial solutions. So as I've mentioned, conventional methods largely use metagenomics approach, or alternatively enrich viral genome by adding more steps. The enrichment part usually adopts one of the two methodologies for targeted RNA-seq. First is PCR-based amplicon sequencing, which has been a popular method for SARS-CoV-2 sequencing. Second is hybridization-based capture with probes targeting the viral genome. You may be more familiar with that due to similar targeted sequencing applications in human exome sequencing, like things like that. The metagenomics and the targeted approaches share the same first step of converting RNA to cDNA via random hexamer primed RT. In V-seq, we perform targeted viral genome sequencing by multiplexing virus-specific primers at the cDNA synthesis step. We designed densely tiled RT primers across the SARS-CoV-2 genome with special design at the 3' end to minimize mispriming from human RNA, which will be the focus for the rest of my talk. Protocol-wise, here we show comparison between the commercial solution using NEB kit versus V-seq. Both methods start with the extracted RNA from NP swabs of COVID-positive patients in the clinical lab. Gray shades are common steps between the two protocols. Other matching colors shows equivalent steps that serve the same purpose between the two protocols, but using different methodologies. For example, NEB, here NEB has RNA depletion, RT with random hexamer, and in the end, some enrichment steps. The same purpose is achieved by V-seq via viral-specific RT. So V-seq enables removing time-consuming steps. We further simplified steps for second-strand synthesis and for adding indexed sequencing adapters. The resulting protocol can be completed within 5 hours from extracted RNA and easily scales to 96-plex index libraries and can potentially multiplex thousands of samples with automation. The simplified workflow can reduce library prep cost by 15-fold and is quite cost-effective. The key steps here is viral-specific RT and technical optimizations around that. So now I'll talk in more detail about the design. So unlike PCR or hybridization-based enrichment, RT primers are usually less stringent and tend to misprime by the 3' end hexamer. As you know, for PCR, we design a primer that's quite long and play with annealing temperature because the entire sequence of the primer actually matters. For RT, the 3' end hexamer matters the most. In human, 90 to 99% of the total transcriptome comes from the rDNA repeats. In 2009, a not-so-random hexamer design was published to eliminate human rRNA from the total RNA by using a subset of 700 hexamers depleted in rRNA genes among the 4,000 possible hexamers. It was used for analyzing pathogen transcriptome like malaria. I'll be referring to this as an SR-seq for not-so-random hexamers. But the enrichment by NSR-seq is still a variant of the metagenomics approach and doesn't enrich over human RNA Pol2 transcriptome. Basically, you're still sequencing a bunch of human RNA. Malaria has a 24 megabase genome, so maybe that's okay. SARS-CoV-2 is only 30 kb, so NSR-seq alone is not ideal as the amount of protein coding sequences in human is still 1,000-fold over the SARS-CoV-2 genome. There's also the much larger sequence space in the human genome, as RT enzymes can also use DNA as template. We therefore adopted an NSR scheme in the design for the 3' end hexamer, but still use the full 25 mer for targeting RT primers. We further expanded the idea to reduce human RNA Pol2 transcriptome by selecting primers with 3' hexamers underrepresented in the human ProSeq dataset bioinformatically. Here's how we design the primers. So first, extract all unique 25 mers in the reverse complement sequence of SARS-CoV-2 genome as candidate RT primers. Filter by GC content between 40 and 60%. Filter by hexamer at the 3' end, where they must be either in the NSR-seq primer hexamers to avoid RNA, or in the bottom quartile in human ProSeq dataset in terms of hexamer abundance to avoid human RNA Pol2 transcriptome. We then remove perfect match to human genome and pick a subset of evenly spaced sequences across the SARS-CoV-2 genome. So we ended up with 390 primers for the first version. Here's how the genome coverage looks like. X-axis is coordinates, y-axis is per primer depth. First point is that the pattern looks holds across different samples, reflecting per primer efficiency rather than sample variation. Second, these are on log scale, so the depth can vary by 100-fold. Blue dots are well covered, red dots are poorly covered regions. They also tend to vary by region. In these two examples, the 1 kb at the top is well covered, while the bottom one is 10 to 100-fold less covered. So we thought to improve coverage in these regions by simply adding more primers and see how it works. Then the question is, can we just solve the problem by adding more primers? Optimistically, as long as RT primers don't interfere with each other, we can densely tile them across the genome bases apart to improve sensitivity. Reverse transcriptase has active RNA-DNA hybrid strand displacement activity. Therefore, by tiling RT primers, each base pair of the viral transcriptome has multiple opportunities to be converted to cDNA in a single reverse transcription step. That's what we tried, and it basically works. I want to make two notes here. First, although we can tile overlapping primers for enrichment in amplicon sequencing, they need to be separated into non-overlapping pools, because otherwise shorter overlapping products will be preferably amplified. Like here in the Arctic design that's popular for sequencing COVID genomes, each amplicon is 300 to 400 base pairs, and the two adjacent pair here, blue and orange, overlap by 60 base pairs, but they have to be separated into two different pools because otherwise they will just amplify up the 60 base pair products and lose coverage. So there's a limit of number of primers you can have because they need to be non-overlapping in the PCR reaction. But for the RT primers, you kind of can tile as many as you want, assuming they don't interfere with each other. Second, enriching by the single-ended RT primers can accommodate highly degraded clinical samples, as we do not require both ends to be present in the same amplicon, unlike in PCR. So we ended up designing these 4 primer pools OP1 is what we initially have with the 390 primers, and the per primer depth has peaks and dips, as I showed you. OP2 is the Arctic design that the COVID community has been using. It has 109 primer pairs designed for PCR enrichment, as in amplicon sequencing, and like here, used in 2 separate pools for overlapping primers. The reverse primer in the Arctic design can be used as RT primers, and they don't really have the NSR or PRO-seq design to avoid human reads from the 3' end. OP3 is designed to complement OP1 for those poorly covered regions. So we've inserted primers in those dips I just showed you. OP4 is the one that uses the most primers from the intersection of both the NSR and the PRO-seq hexamers, so we try our best to avoid both RNA and human transcriptome. Now I'll finally start to show you some results with these primer pools and their combinations. So here we show viral genome coverage with at least a 5x depth as a function of CT. Overall, with 90% coverage as the cutoff, we recovered 94 and 71 successful genomes with a median depth of 700 and 700x and 13x by V-seq and NEB, respectively. The contrast in total raw reads and the median depth reflects an average of 90-fold enrichment of viral reads by V-seq compared to NEB. Although I need to mention that we've attempted more high CT samples with NEB, so this is not an entirely fair comparison. Later, I will show you fold enrichment of viral reads as a function of CT. Both methods robustly work for samples of CT below 24 with greater than 90% success. Such CT cutoff covers 2/3 of all the positive samples at our clinical lab. For the rest of the samples with CT greater than 24, we had sporadic success with NEB in an or a non session. We either obtained greater than 90% coverage, which is about 16% of the samples, or we almost entirely failed to obtain any viral reads. V-seq is a bit more complicated. Here's a further breakdown by primer pools. Basically, more pools combined, more primer used. With the optimized primer pool combinations, which we use about 800 primers, we attempted with V-seq on 5 samples of CT between 24 and 25, as well as 1 sample at 34. 2 samples have greater than 90% coverage. All the rest had intermediate coverage between 15 and 78%. Here we compare primer designed with NSR and PRO-seq hexamer ends with Arctic primer in an internally controlled manner in terms of genome coverage. Basically, we separate out the OP2, which is the popular Arctic design people are using without the NSR and PRO-seq design at the 3' end. We found improved performance of our primers, primer sets, because partly due to that they cover the genome with increased number of primers and better on-target specificity. But most importantly, denser primers do not interfere with each other, but act rather synergistically to improve genome coverage. Because otherwise, more primers don't necessarily help. This is sort of the take-home message from our optimization. Although results in the clinical samples above suggest that densely placed primers improve genome coverage, particularly as CT increases, each dot is a different sample, so it's not the kind of well-controlled experiment we are more comfortable with that are done on replicated samples. So we further evaluate this trend on the same sample, but diluted 1 to 10 as replicates and downsample the sequencing reads to the same depth that doesn't saturate the whole genome. We diluted 2 samples and tested 9 primer pool combinations. Consistent with the trend found in clinical samples, increasing the number of primers improve coverage, particularly for sample dilutions with higher CT, which are the ones with red and orange dots here. So here we show percentage of on-target viral reads and fold enrichment as a function of CT. At lower CT, you get almost all viral reads, but it's also not bad with NEB. So below CT of 20, you get about 10-fold enrichment. From CT 20 to 25, it gets more helpful with V-seq, and you get about 100-fold enrichment and occasionally several hundred-fold. This is showing percent viral reads in the diluted samples, breaking down for different primer pools. As as expected, the primer pools that include OP4, which consists the most 3' end hexamers from the intersection of NSR and PRO-seq hexamers, perform slightly better than the pools without it, because OP4 most effectively excludes contaminating human reads. So improved viral genome coverage simply by adding more RT primers suggests that overlapping primers do not kinetically interfere with each other in their binding to the RNA template. Here we look more carefully into how overlapping primers behave. We show positions that are primed more than once. X-axis is coordinate, y-axis is the number of times a position is covered by designed primers. We only show those covered at least twice. In other words, by overlapping primers. In the original OP1 design, 27 out of 390 primers have 3' end located within 25 nucleotide of the preceding primer. Since the primers themselves are 25 nucleotide in length, we call these overlapping primer pairs. In OP134, where we combined almost 800 primers, where we added primers in those like poorly covered regions, 42% of the primers overlap. So we then examined all the overlapping RT primers and compared their performance with randomly sampled primer pairs from all the non-overlapping primers. Here we analyzed the ratio of median depth for primers in an overlapping primer pair or in a randomly sampled pair. We take read counts for the first primer in pair, which is the preceding primer at the 3' end of the RT, divided by read counts for the second primer in pair. We expect this ratio to be varying around 1 when they do not interfere with each other. As expected, this ratio is not significantly different whether the primer is in overlapping pairs or not. We then analyzed the average median depth between the primers in an overlapping pair or in a randomly sampled pair, primers within 25 nucleotides on the left and within 6 on the right. The average efficiency between the two primers is slightly reduced in overlapping pairs within 25 nucleotides than in randomly sampled pairs, and not significantly different when evaluated for primers within 6 nucleotides. Most importantly, we evaluated whether having multiple offsetting primers help recover otherwise sparsely covered regions. Here we show that with overlapping primers, we increase depth for 37 poorly covered regions by 2.7-fold with an improved minimal depth of 28. These poorly covered regions with depth less than 10 by a single primer are largely recovered now by optimized primer pool. So to sum up, V-seq provides cost-effective sequencing for SARS-CoV-2 with 15x cheaper for library prep and 90-fold enrichment for on-target sequencing reads. I didn't mention that V-seq robustly calls identical variants with NEB. It works well for samples with CT less than 24 and provides intermediate genome coverage useful for viral strain analysis for higher CT samples when full coverage cannot be obtained due to low viral load or sample degradation. The protocol takes about 5 hours from extracted RNA, which is much shorter than commercial kits. Beyond this specific application on SARS-CoV-2, I'd like to quickly discuss a few additional implications of relevant applications. First, NGS-based COVID testing, like swab-seq you heard last week, has been developed to scale up testing. In a nutshell, swab-seq uses 1 or 2 viral-specific RT primers with sequencing adapters and converts conventional RT-qPCR readouts of CT to normalize the viral read counts. Like metagenomics, amplicon sequencing, or hybridization-based methods, like those traditional methods, share the same random hexamer primed RT, such that the effort of testing versus whole genome sequencing is bifurcated at the very first step, with viral-specific RT for testing and non-specific RT for genome sequencing. V-seq, however, naturally integrates with NGS-based testing and can potentially inform viral strain concurrently with testing. Meanwhile, V-seq can also be leveraged to select efficient and specific testing primers to assess their cross-reaction in a pooled manner and to include other pathogens like influenza. V-seq can also serve as an optimization platform for other amplicon-based diagnostics or genetic testing, for example, to analyze pathogenic non-coding RNA or splicing errors. Lastly, viral genome sequencing with matched CT provides an interesting platform to optimize sequencing for targeted transcripts with abundance varying across 4 orders of magnitudes. We learned that densely tiled arch primers improve coverage and that overlapping primers act synergistically for recovering difficult regions. Such knowledge could help with overcoming technical difficulties surrounding targeted RNA-seq. In some way, anticipate that V-seq can be applied to cost-effectively investigate genome evolution of RNA viruses and to other research involving targeted RNA-seq. So with that, that's all, and I'd like to thank the people involved. It will just be a protocol on paper without all the work by people on this page. First, Long did all the experiment. He's a postdoc in Leonid's lab. James and Josh helped with the pre-processing of all the data. Josh and Leonid co-supervised the work. And I'd also like to thank Leonid, Jonathan, and Eliasa for involving me in the COVID research in the first place from late March and being extremely supportive in terms of the resource and help. Sean and Omai for providing the clinical samples. Jacob was my previous lab mate in Jay Schindler lab and a guru in targeted RNA-seq. Yi, John, Laila, Chongyuan, Shui, and Valerie for help with shuttling the samples, purchasing reagents, and providing helpful advice. And Ximin and everyone in the sequencing core for accommodating all of our sequencing rounds in 1 day notice. Thanks for your attention.
Thank you, Yi. If there's no burning questions in this moment, we'll take questions all together at the end after Leonid's talk also.
Okay, thank you, Melody, and thank you for an extremely generous and kind introduction. This is actually, I realized this is my first time actually giving a Zoom talk. I've been in an enormous number of Zoom meetings since March, but this is my first time trying to present without being able to see all of you, so bear with me. And I should say at the outset here that I'm not a virologist, and this is really my first venture into viral phylogenetics. So I guess by way of background, I think as Yi kind of mentioned, when UCLA was starting to ramp down in mid-March, a number of us on the basic research side got together and decided that we were interested in seeing what we can do to use our genetic and genomic expertise to help fight the pandemic. And we got connected with Omai Gardner in the clinical lab, who you heard from last week, and launched what's been kind of a series of very fun and productive collaborations. So you heard last week from Val Arboleta about our efforts to develop a new, much more scalable, inexpensive, etc., technology for testing for SARS-CoV-2, which we envision as a way of attacking the pandemic with very widespread population screening for the virus. But the other thing that when we started talking with Omai, he said would be really useful for him and his group, as well as more generally, is for us to sequence the viral genomes and look at kind of the molecular dynamics of the pandemic locally in Los Angeles. And a number of such studies have been done, before us and contemporaneously with us from all over the world. So we can draw on that information as well, but hopefully shed some new light on what's been happening locally. So I'm gonna share the slides here. All right, here we go. So I want to talk about using both of the technologies that Yi just described, the standard commercial NAB technology, and then the V-seq technology that she developed to look at viruses in LA. And I'll thank everybody at the end. So as you heard from Yi, V-seq is a highly accurate SARS-CoV-2 sequencing approach that's a lot cheaper and more efficient than the NAB method. And what we really care about in this epidemiology project is making accurate variant calls. So sites that are not the same in every single virus, but that distinguish viruses from one another. And that's the information that we're going to use to figure out their history, common origin, where they come from, etc. So, okay. So we care about the quality of these variants, and there are 18 samples that were sequenced successfully using both NAB and V-seq, and we compared 379 sites across the SARS-CoV-2 genome at which we saw sequence differences, and calling which allele was present in each given sample gave us over 6,000 high confidence genotype calls, and we really saw no discrepancies between these two methods, as illustrated for just a couple of sites and a couple of samples here. Yeah, so in total with the two methods, we successfully sequenced 180 genomes, and our standard for successful here was at least 3x coverage for at least 80% of the genome, and most of them are much better than that in terms of both the depth of the coverage and the extent of the coverage, but this is the minimum level where using some computational tricks we could rescue enough variant information for the genome to be useful for what we want to do with it. And these correspond to 162 unique patient samples from the clinical microbiology lab or my lab at UCLA Health. So a little more than half of them were done using Yi's new method. So what have we learned from looking at these samples? So what we did is we took these 162 unique genomes sequenced at UCLA, and we combined them with 144 publicly available genomes sequenced by a variety of groups from LA County. So that's 306 LA County genomes. And then we clustered them with over 4,000 genomes sequenced by groups around the world and publicly deposited in this public GSAID database. I know this is kind of a hard plot to look at, but in purple, so all the black triangles are the LA samples, and then in purple and mostly seen exclusively down here are the Asian lineages. So this is the original like Wuhan lineage and its close derivatives. And then in green are the subsequent strains that appeared in Europe, I guess first in Italy, and then spread to New York and North America. Blue shows strains that have been characterized particularly in North America, and most of them form a subset of this large green clade. As well as a few down here in the Asian clade. And what sort of obvious thing you can see here qualitatively is that the LA genomes are distributed throughout this tree. So there's some that look a lot like the early Wuhan strains, this guy in particular we'll talk about more, yeah, as well as strains that look like they came from Europe, and from other parts of the US. So as might be expected for a major, very cosmopolitan global city with one of the busiest international airports in the world, there have been lots and lots of introductions into LA, and they came from anywhere the virus was circulating in large numbers. So we wanted to, so kind of the rest of this is going to be going through this in a little bit, with a little bit more granularity. So again, the black triangles here are the LA genomes, and the red asterisks or stars are computationally inferred original progenitor strains for all of these LA genomes. So by looking at some of these 300 odd genomes, we identified what looked like 167 separate introduction events. And only a minority of them, 35, resulted in observed community transmission, which means that we saw multiple samples with that particular lineage, going back to that origin. And as has been seen elsewhere, with people have kind of said, like, there are a lot of embers being thrown off by this pandemic, and they land somewhere, and most of them extinguish, and a few catch and grow into big fires. One of these introductions is responsible for a significant fraction of all of these cases, right? So 66 of our genomes go back to this one introduction event, and for most of the apparent introductions, they're singletons. So we see no evidence of community transmission. Although, of course, what we're looking at here is only 300 out of probably I don't know what it is now, 300,000 or maybe up to 1 million instances of SARS-CoV-2 infection in LA County. So some of them may have given birth to much smaller to smaller clusters that we simply don't resample multiple times. So again, to kind of illustrate the point of widespread global introductions, if we look at what's been coming into LA, and this is only looking at February through about the first half of April, so kind of what we think of as the early phase of this pandemic, you see lineages that are characteristic of lots and lots of different origins of SARS-CoV-2. So you see the Wuhan strains here in light purple. You see the strains that were characteristic of Italy in blue, strains that then were seen in Italy and really went wild in New York, and there were a lot of them getting introduced into LA in March and early April, as well as Iran, Northern California, Washington State, etc. But what we see here in LA and what's been seen in many other parts of the world is that the B1 lineage and its derivatives, so this is a lineage that distinguishes the Italian outbreak from the original Wuhan lineage, and its derivatives have been introduced to county since then. And we really don't see any trace of community spread of the older Asian and other lineages that we were seeing at the earlier dates. So as has been reported, these B1 lineages carry this famous D614G mutation in the coding part of the spike protein, the gene encoding the spike protein. And it's been argued that while it doesn't make the virus more dangerous, more pathogenic, it does make it more transmissible. You can argue about how persuasive that evidence is and what the balance is in terms of the possibility that it really is spreading more rapidly and taking over for that reason, or that it just got lucky and spread through genetic drift. But as has been reported elsewhere, as the pandemic progressed, we watched these lineages take over everything else, even though we can clearly see that a lot of other stuff was circulating during the early stuff of the pandemic, during the early part of the pandemic. Okay, so basically the rest of the talk is going to be about one particular lineage or one particular sample and the lineage or lineages that it's been involved with. And this is a sample that was collected here at UCLA on February 21st. So this is quite a bit before they were actually doing any COVID testing. So the sample was collected as part of flu testing, and it tested negative for the flu. And then a project that Evan and others have been leading in the clinical lab has been to go back to some of the flu samples from January, February, early March, and retest them for SARS-CoV-2. And so indeed, this sample tested positive for SARS-CoV-2, and it carries this D614G mutation in the spike protein. that distinguishes the later viruses that have taken over from the strains that were circulating earlier. And I'll note here that this is the earliest European lineage that has been seen in the US to the best of our knowledge. It's not the earliest lineage that's been seen. There are some strains from Northern California and Washington State that go back to January, but all of those look like the original Wuhan strain. And this sample was isolated in LA in February 21st, which is 18 days before March 11th, when the travel ban from Europe was instituted. So by that point, the cat was definitely out of the bag already. This is an interesting lineage, this B143. It appears to be US specific. And here's where it's been seen in the US. So there are 101 instances of this lineage that were found in California, a number of them by us in this study, 54 in Washington State, and then there are 5 in Utah, a couple in Arizona, and just singletons in a few other states. And it's not really seen anywhere else. So it's not quite unique maybe, but presumably originated and is almost entirely observed in these states. And I'm guessing what we see in other states is travel from here. So out of the 186 B143 lineages that have been identified worldwide, 101 were found in LA County. So that's 48 of them, about half, and other parts of California. So it's very common in Contra Costa County. Blue here is this lineage versus orange, all the other lineages found in those places. But really all over California. There's a center in LA, and in parts of Washington State, but these are not the Seattle isolates, which tend to look like Wuhan and other strains, but Whatcom County and Skagit County in northern Washington State, where this lineage really is predominant, and possibly where it originated. So there is an interesting travel history associated with the sample that we identified this lineage in. So it was introduced once into LA County. It's the source of the largest cluster that we've seen, the 66 genomes that are the biggest identifiable community outbreak here. And this sample is the earliest B143 sequenced. And this particular patient, going to the travel records from when they were seen, traveled to Seattle on February 15th from LAX to visit family, came back 5 days later and tested positive the day after that. So one could guess that they went to Seattle, picked it up there and brought it back. It's also, I guess, possible that they had it before they left, and that they're the ones that introduced it into these parts of Washington State. And I guess one thing I'll close with, which is a little bit speculative, is that in this sample, in this particular sample, we actually see what look like multiple viral genomes. So when we look, there are a number of sites where we see within patient variability. So we'll see, lots of reads, lots of high quality sequences, and some of them have this AAC sequence that deviates from kind of the common reference sequence, and others are reference, GGG at these three nucleotides. So, obviously this is the sort of thing that, maybe you don't expect to see, and you might worry about arises from any number of, mix-up contamination events, data analysis events. And we've looked really hard at that. And from everything we can tell, including samples that have been sequenced multiple times by multiple approaches, it looks consistent. It looks like this sample really did have a mixture of two viruses, but we can't, of course, absolutely rule that out without somebody having gone back and sampled that patient again independently at the time that they were infectious with this virus. But we looked in other samples and we had identified these possible signatures of co-infections in 10 other patients. And they're not like supported by a single, by a single site. They're supported by between 5 and 16 variants, which is kind of comparable to the number of variants that distinguish strains more generally. And this particular co-infection of B143 and B11 that was seen in this patient, in this earliest LA patient, is also found in 6 other patients. So we see a total of 7 other samples where it looks like it's a mixture of these 2 genomes. And when you look, it looks, all of them are found early. So this LA0193 is the earliest of these samples, and it looks like the ancestor of all samples in LA that are B143 and all samples in LA that are B11. Early, you see, so these red lines here connect to what look like 2 different genomes that were found in the same patient. And it looks like early on, there was transmission of both of them going on. And then at some point, they got sorted out. And now, more recently, you see either the B143 genomes or the B11 genomes. And we've stopped seeing patients who share both of them. So that's kind of an interesting and unexpected observation. So I'll conclude here. So as Yi mentioned, Long in my lab did all the hands-on work for the sequencing. It was great help from Zimin at the sequencing facility, and they really went above and beyond there to prioritize our samples, because some of this was also of interest to the clinical lab to track specific outbreaks in LA. James, a student in my lab, did all of the analysis. Yi and Josh supervised the molecular and analytical work, and Yi, of course, is the originator of the V-seq approach. Jonathan, Eleazar, and Val have been great co-leaders of this whole team effort to try to use genomics and genetics to fight SARS-CoV-2. And then Omai and Sean and Evan and others in the clinical lab have been absolutely instrumental in this. We obviously couldn't have done any of this without them and their careful collection of these samples, ability to go back to them over time, even before there was testing, etc. So I'll stop here, and I guess we can all take questions.
Great, thank you so much to the speakers for really, really interesting talks. I think there are some burning questions already. So I think Manish Butte wants to ask the first questions, and then it's Owen.
Great.
Go ahead, Manish.
Thanks. Great talks. I learned so much, and I thought I knew so much about COVID, and I keep learning. This has been wonderful. Thank you so much. Yi, I want to ask you a quick question about when you do RT from, in both directions, you might be picking up the negative strand. And correct me if I'm wrong, I'm not a virologist, but for positive strand RNA viruses, detecting negative strand actually implies replication of the virus. Can you use a detection of negative strand on the RT and then subsequently sequencing identification of the opposite strand, the complementary sequence, as evidence of replication, of ongoing replication of the virus?
Yeah,
infectiousness
sorry,
of the
it wasn't clear. Yeah, so all the primers are targeting the positive strand. So yeah, so we didn't include the ones targeting the negative strand. That's like one super interesting point. But for the NEB-based experiment, they are coming from 2 strands. So, but in terms of the if you track the variants, they should be, if you are thinking like RDRP, like the replication process of the virus can introduce variants, but those heterozygous variants are not in both, they are in both methods. So yeah, I guess it's also related to how you collect the samples, because I assume the replicated samples should be within a cell, But when people are doing the testing for the NP swab, if you spin down the cells, they are there's very little things in the supernatant. So I mean, there's a whole bunch of virus in the supernatant. If you spin it down, it doesn't use the pellet, it doesn't increase the viral load. So I think what we are picking up is more likely to be the packaged virus, but I'm not a virologist, so I'm not entirely positive. Yeah,
Okay.
but I think with the NEB, we can look into the negative strand. That would be super interesting.
And then the question for Leonid, I really love that. And I was wondering if you might, since you've analyzed all many samples, there have been around 350 or so UCLA health patient workers who have tested positive. I'm wondering if you're able to discern whether those workers got it from the hospital patients or whether they got it from each other healthcare workers based on the sequences. Like, is there an outbreak within UCLA Health, within a clinic, et cetera? I guess.
Right. So that,
you get what
yeah,
I'm
so that was kind of why Omar and Sean and Evan and those guys were interested. You know, this was like a super high priority project for them. And I know that they're looking closely at that. So maybe, I think, I mean, I think, yes, you can see all of those. You can see some of those things, but I'm not sure exactly what the answer is. And I kind of don't want to steal their thunder. So, but that was partly, I mean, that was kind of the main motivation from their side, was exactly for that reason. And we really prioritized the samples that came from the healthcare workers.
Okay, thank you.
Yep.
Great. So the next question is from Owen Whitty. What aspects of fitness or enhanced pathogenesis can be answered with your data and approach?
Yeah. So actually, can I just amplify on the question before they get a chance to answer?
Oh, Owen, take it away.
So thanks. I really enjoyed the talk. So this issue of fitness and selection of, for outcompeting other strains of viruses is pretty important, particularly if we're going to have, a chasing target in terms of drug targets or immune targets. So what have you learned about these different substrains that have been identified in terms of dominance in their region? So, thanks.
So, I mean, I don't know that we've really learned anything about that. I mean, we can confirm what others have reported, which is that this D16 614G seems to take over. It took over here just like it did everywhere else. It is controversial to what extent that's evidence of selection, right? Because it could also be that just by dumb luck, that strain was in a super spreader early on in Italy and basically seeded that entire outbreak. And then those are the strains that jumped to New York, and that's why you see them everywhere. People have done some, the group that originally proposed the hypothesis that it's more infectious, they did some cell culture studies that they claim were consistent with that. I mean, that's not my expertise at all, but having read like all the commentaries on that work and stuff, people weren't entirely convinced that what was being reported was meaningful to, human to human transmission in a realistic natural setting. So I think it's still controversial, and you'd need a good animal model, and you'd have to figure out exactly what the right animal model is, kind of like they use ferrets for the flu. And obviously, you're talking about BSL-3 and all of that stuff that we that we don't do. I guess the other thing is, from the folks who've looked at this structurally, it's not thought that this particular amino acid change would affect antibody binding. And so in terms of vaccine and neutralizing antibodies and all of that stuff, this one shouldn't throw off the efforts. And it's really the only variant that's been found so far for which there is even circumstantial evidence that it makes the strains functionally distinct, as opposed to just random SNPs that help you do the phylogenetics but don't do anything to the virus.
Okay. So the other question, which you may have covered, and perhaps I didn't catch it, did you find significant variation within the polymerase and anything that might correspond to resistance mutation for remdesivir?
Not that I know of. We can go back and look more specifically, but I don't think I think if that had been seen, I think we're seeing all the same variants everybody else is, plus a few more that are private to some of these lineages, right? I think it would be known, but we can look again. That's
Okay.
good.
All right. Thank you. Appreciate it.
Great, and then we have a question that seems to be answered already from Joseph at Pisagna. Maybe the speakers can add more. What are the major differences between B.1 and B1.143?
Yeah, so I think as James notes in the chat, there's 7, he thinks, SNPs that distinguish it. And these are all D614G strains, so functionally, functionally, they should be the same as far as anybody knows. So it's just presumably, as like for all of these other strains, as they diverge occasionally, there is a replication error, and you pick up a variant that allows you to tell the lineages apart and kind of track the geography and the passage of time. But that's about it.
Leonid, to follow up on Owen's question. Are there any differences in the S protein, the region that encodes the S protein, mainly because, so at the VA, we're about to start the Moderna trial, which is the vaccine trial. So it would be, I think, of huge interest whether there's any variations in those regions that we're using in our vaccine.
Yeah, so as a spike, right? So that D614G is a spike mutation, which is why it's been so interesting. And I don't remember exactly where it is with regard to like the ACE2 binding site and all of that stuff. But I think people have looked at the structure and don't think that one amino acid substitution would affect vaccines. It's certainly something you could track, right? But it's probably largely of academic interest at this point, because essentially all strains all infections you're seeing now have that mutation, the vast majority of them. So it's not like, you could really stratify your trial by who does and who doesn't. You'll just see, if you're recruiting now, everybody with that variant.
Great, we have a hand raised in the audience. Roel, if I didn't butcher your name, I apologize.
That's okay. That's okay. Don't worry. Great work, by the way. Thank you so much for presenting. It's great work. I really appreciate what you guys are doing. I have 2 simple questions. One is you mentioned the co-infection of multiple strains, and I wondered if there's any other evidence from, let's say, the common flu viruses that this phenomenon has been seen before, and whether or not how novel this is. And the other question I have is different. Is this: does your data provide insight in the mutation rate of this virus, the coronavirus? The mutation rate. People have been talking about changes that we should face in the next few years, and how that impacts, for example, vaccine development. But does your data provide any insight there?
Sorry, what was the first part of your question again? I started thinking about the second part.
Yes, there's always the danger of asking 2 questions. The first one is about the co-infection. You still found multiple strains. And I wonder if it's seen before for, let's say, the common flu.
So like I said, I'm not a virologist, so I don't actually know. I know for like HIV, right, which has a much higher mutation rate because the polymerase is super noisy. They're totally like quasi-species within individuals that are very different. And then there are bottlenecks during transmission events and all of that stuff. With the flu, I don't actually know. Melody, do you know?
The thing with flu, again, I'm not a flu expert, but the thing with flu that's really interesting is, usually in the population, there are multiple strains circulating. So you probably heard about, I don't know much about within the person, but thinking more about within the population, one unique feature about the flu is that when the swine flu came out, it completely displaced the seasonal flu, I believe. And I think there's very complex dynamic, like kinetics there, that determines what strain predominates. But I would say, like what Leonard was saying, that we regard basically all RNA viruses with error-prone polymerases are capable to generate this quasi-species. So usually within different tissues or individuals, you're looking at a population, a galaxy of viral mutants. But there is exclusion of superinfection, meaning that for some viruses, infection by one virus strain could prevent infection by a new one. So that is also seen for some viruses.
Yeah, so, and in terms of the mutation rate, I mean, so yes, you can use this to infer a mutation rate. No, I don't know what the number is in, you know, base pairs per day or whatever. But I guess, like, crudely, right, the genome is about 30 kb, and they're about 300. James says it's about 2, you know, substitutions per month per lineage. So 20s per year, right? So we see about 300 variable sites across this whole tree, and the genome is about 30 kb. So about 1 in 100 bases are seen to vary in the population in this at this point.
Any other questions?
Just one quick, so if you have a multiple strain infection, that would suggest that the person acquired it from like a super infection event, or like from multiple sources, like being sort of in a closed room with multiple different infectors. So these results, I think, are really important epidemiologically, because it would help you do contact tracing from either a large group event versus a single source like a healthcare provider. I think Manish asked earlier about healthcare transmission. So if you're a healthcare provider and you only get a single strain, it would suggest that you're getting it from a patient. Whereas if, let's say, that healthcare provider had multiple strains, it would suggest they probably got it from a group setting. Those are just my thoughts.
Yeah, and I mean, if I had to guess, right, given that we saw this early in this one sample and a few others, and then we saw transmission from this sample. I don't remember exactly what the number is, but, you know, obviously we're not going to see it if the admixture of one type is very small. So this is somebody who is carrying whatever 70% of their virus is one lineage and 30% is the other. And every time they transmit, there is some noise about that, and eventually one or the other of those lineages is lost along the transmission chain. So that's what we see. But yeah, I mean, if I had to guess, the fact that we saw this early, I would guess that this was probably, something that occurred in a healthcare setting where somebody was exposed to multiple infectious patients before people figured out all the PPE and all the other stuff and how to deal with this properly. That would be my guess, because yeah, you would have had to have been exposed to a significant dose from multiple individuals who acquired it from different sources or something like that. So hopefully that kind of stuff isn't happening as much as we've all learned to be more careful in what we're dealing with.
Yeah, I hope to add from the technical perspective, like these are from NP swabs mostly. So if there are co-infection, they need to be at high dose in the nose, like to be picked up. But I think there's like from the clinical lab, they saw that sometimes the viral load in the nose will decrease, and then very few of the samples are from the lavage. So that might have more like co-infection detected because both will be there. So I guess like, so for us to detect a co-infection, it sort of needs to be at the nose, at the same time.
Great. I was wondering if I can ask one more question about the co-infection. We know that for coronaviruses, recombination is a major mechanism for diversity. So I'm curious, in the co-infected individuals, can you do like nanopore sequencing, for example, to see if there's actually new recombinants that come out from the event?
Yeah, so we've looked a little bit at using nanopore, and I mean, in theory you could. In practice, I don't remember how far we got, but I mean, what we did was computational phasing of these different genomes, and we have not seen, unless James chimes in and tells me otherwise, evidence for recombination. It really looks like it's kind of all one lineage and all the other, which doesn't mean it can't happen, right? But I mean, we've only seen a handful of examples where there are, you know, two and only two lineages. So, and if there was a rare recombinant between them, we might not see it. But as far as I know, none of the lineages really look like, you know, recombinants of two other lineages that at least are distinguished by enough sites that we could tell, right? So far, it's the whole kind of viral tree is young enough and the mutation rate is low enough that they're just not super different from each other. So there could be a ton of it going on just undetectably, where you swap sequences that are not distinguishable by sequence variation, or that are distinguished by one SNP, and then you can't tell if that's a new mutation or something that got acquired. So so we are aware, and we've looked at least a little bit, but we haven't seen anything that would really argue for that. If you did, you could then do nanopore or something like that and try to convince yourself that it's real.
Yeah, I think so. For recombination, when people talk about it, it could be that it's a recombination in the viral genome. So they are in the virus particle for a whole genome. But it could be, I think mostly people mean it by replication like slippage. So it's like the RNA polymerase RDRP like jumps. So in that case, you would, if you have a recombination, that will be in a heterozygous situation. In terms of the sequencing technologies required to detect it, if you use, even with nanopore, but you need to convert the RNA into cDNA. So that you need to use a reverse transcriptase, and all of those have a pretty high jumping rate between templates. So even if you see one, which we didn't, like it may not be like for sure like that it's if it's in a heterozygous state, unless you see it in a homogeneous population, I guess. But with nanopore, if you do direct RNA sequencing, you can actually see the recombination. And there's a COVID transcriptome paper with direct RNA seq with nanopore that reported some recombination. Yeah.
Great, thank you so much. And I guess with no further questions, that's it for today. Thank you so much to the speakers. We really enjoyed the presentations, and see you next week. I believe next week we have Jason Ernst presenting on Wednesday. So
thank you.
Thank you.
Thank you.